So when you want to identify a particular type of a protein from a mixture of proteins, this Western blotting is going to be helpful. So in this method, labeled antibody against your particular protein is used to identify the desired protein. So it is a specific test. And the Western blotting is known as a immunoblotting also, okay? Because it uses antibodies for the detection. So here you cannot use any probes, right? Because there is no sequence in this case. So here you have to use your antibodies, which are going to be specific, which are going to be, which can go specifically go and bind to your antigen or your protein of interest, right? So these are the steps that you have. Almost the initial steps are going to remain the same. What you're going to change is this treatment, or the detection method is what is the change here. So the first step is the extraction of the protein. Once it has been extracted, again there would be gel electrophoresis, SDS phase to separate out. Then once it has been separated out, then electrical or capillary blotting, or combination of that, what we use today, that can be blotted onto your nitrocellulose membrane. Once it has been blotted, then blocking using BSA, the other, whatever your other positions are there. Then treatment with primary antibody. Then you give a treatment with secondary antibody, and secondary antibody. Yes, Soumya wants to say something? Here you are not doing denaturation, no? You are talking about SDS page. SDS page, because of using of SDSD denaturation. That's how you are talking about? Yes. So, see, if you are using SDS page, you have to use an antibody which would be more. See, antibodies can be again of different types. Okay, there could be some antibodies which can bind to the inner sequences of the proteins, and there could be some antibodies which could bind to only the structures, or you can say only the outer structures, or the what you can say, like your the overall conformation of a particular protein. Okay, so if you are using SDS page, so yes, definitely, if you are using a antibody, depending upon what antibody that you are using, you have to suggest whether you will go for native page or whether you will go for SDS page. Yes, sometimes we go for SDS page, and most of the times we can go for native page, where the conformation of the proteins is going to be restored. Okay, yes. Very good, Soumya. A good question for everyone. So once that is being done, so primary antibodies, and then you have the secondary antibody, which is enzyme labeled antibodies. Now, tell me, everyone, that you must have learned this in ELISA also. You are studying here also. I think I have discussed this earlier with you, maybe when I'm talking about ELISA or anything. So tell me, why do you always require a secondary antibody in this case? Like, for example, here you're going to have a, so let me just present it so that I can, so for example, if you have this as a protein, right? So I can have directly a primary antibody, okay, which can go and bind, and this primary antibody itself can be labeled by your any fluorescent dye, or say any labeling. Why do I have to? So in this case, what is going to happen is your indirect way, right? You have your protein. To this, you have your secondary antibodies added. And the secondary antibody is having the label. Am I right? Everyone clear? So my question is, okay, Soumya is writing, due to conformational change, the dye may not be expressed. No, that's not right. The dye doesn't need to be expressed. The dye is already present. Okay, the dye is only going to give it fluorescence. But something closer, something closer. Yes, anybody? Anybody else? You can unmute also and speak up. Secondary antibodies linked to an enzyme substrate which is on incubation will develop color. Yeah, that's true. But my question is, why not label the primary antibody only with the enzyme, so that when it is incubated with the substrate, it will develop the color? Why you need a secondary antibody for that? It's becoming indirect, right? Why don't we go for a direct method instead of going for an indirect method? That's my question. Yes, anybody else? Anybody else? Okay, so let me answer this question quickly. So for specific detection, yes, one thing is for specific detection, and you can say to increase the sensitivity. So how do you increase the sensitivity? That is the question. So I'll again just show you with the pen. See, what happens is when you are talking about, so this is just a second. So this is a protein. So now when an antibody, a primary antibody comes and binds, so what will happen is there would be only one primary antibody. So usually what happens is there would be one primary antibody which will come and bind to your this particular protein. Okay, or you can say that it has been a kind of a bridging together. Yes, so it usually bridges together, and there would be different two different proteins as a that has been binding, right? But when you use secondary antibody, so what happens is that secondary antibody will utilize this entire. The secondary antibody is not only one type of an antibody. There would be many antibodies which is going to be specific for this entire primary antibody. So now what happens is to one primary antibody, you can have many secondary antibodies binding to it. Okay, so many secondary antibodies will bind to it. So what is going to happen is, so just an example over here, I have shown you that there are say one primary antibody and that is binding to 5 secondary antibodies. So the reaction which would be there, it is going to be for 5 enzymes that are being present. So more amount of your product is going to be formed, which would be detected easily, which could be detected easily. Yes. So that is one method. Specificity, sensitivity is increases. So even if your protein is very small in number, very less in number, less in concentration in your sample, still that can be detected. Okay. Second reason, when you are labeling this antibodies with your enzyme, yes, there are changes in the, there could be some changes in the secondary antibodies. Okay. That may affect the binding it to the primary antibody. Yes. So if you are labeling to the primary antibody itself, then the labeling or binding of your antibody to your protein itself is going to be changed. Yes, or the binding may not take place. So that is the reason why always a secondary antibody is being used. Always you go for your indirect binding as compared to your direct binding. Is it very much clear, all of you? A quick yes or no? Okay, good. So let me move forward. So treatment with specific substrate, like you have alkaline phosphatase is the enzyme. Then the mostly it is the enzyme that is being used in these methods. Then substrate is your paranitrophenol phosphate, and which will develop a yellow color after the phosphatase has been bringing down or cutting down your phosphate. Okay, so this is your entire thing. The same thing what you have seen, that whatever the proteins that has been isolated, so that would be loaded onto your SBS page. It will be running, and then once it has been running, then you can have like this transfer from gel to the membrane. So this is this is the blotting method where a vertical method is being used. So this particular diagram, what you see, is only for the ionic transfer. Here, your mostly your blotting or that capillary action is not being utilized. It is a vertical chamber. ऐसा vertical chamber होता है जहाँ पे जेल को तुम ऐसे रखते हो. तुमने जो chamber देखा है horizontal chamber देखा है जो हमारे पास है in our college which we have, that is a horizontal chamber, and those chambers are useful when you want to club ionic transfer as well as your capillary transfer together. In vertical chambers, it is only your mostly your ionic transfer that takes place. So once this transfer has been taken place, the blocking would be done again by milk casein or BSA. Then incubated with your primary antibodies. You can see over here how your primary antibodies go and bind to your protein of interest. And once that has been done, your secondary antibody, which has been labeled with your HRP, is going to be produced horse radish peroxidase or alkaline phosphatase, any of the enzymes to be done. So depending upon your antibodies, then you would be having your whatever the enzymes are there, that enzymes are going, substrate is going to be exposed so that there would be such kind of your bands which are going to form depending upon what is the or where is your protein of interest located, right? Such kind of spots तुम्हें मिलेंगे when you are seeing your results. Okay. Okay. So quickly let us go through these steps. So first is your extraction of protein. So cell isolate is more common sample for Western blotting because it will give a complex or what do you say, mixture of your proteins. Protein is extracted from cells by mechanical or chemical lysis. Okay. To prevent denaturation of the protein, protease inhibitors are used so that the proteins are not getting denatured. So the concentration of protein is determined by spectroscopy. When sufficient amount of protein sample is obtained, it is diluted with loading buffer containing glycerol so that it can sink into the well. Tracking dye, that is bromothiamyl blue, is added so that you can monitor the movement. Then you have gel electrophoresis. I'm not going into the gel electrophoresis in depth. In the next lecture also, we have to see about this particular gel electrophoresis. Then you have the blotting method, which is similar like your northern and southern method. So nitrocellulose membrane is placed on the gel. Separated proteins from gel get transferred by capillary action. So only by capillary action, it may take 1 to 2 days. So to make it more efficient, we have electroblotters where electricity is being used. So ionic transfer is also getting used up. So in electroblotting, nitrocellulose membrane is sandwiched between gel and cassette of filter paper, and then electric current is passed through the gel, causing transfer of proteins to the membrane. Next is blocking. So very important step. So where the nitrocellulose membrane will block the other parts so that the antibodies don't go and bind to other parts of your nitrocellulose. Then you give treatment with your primary antibody so that antigen-antibody complex has been formed. Then you give treatment with secondary antibodies which are labeled with enzymes like alkaline phosphatase or horse radish peroxidase, and then you interact with the suitable substrate. So visualize the enzyme action. The reaction mixture is incubated with specific substrate. The enzyme converts the substrate to visible colored product so that band can be seen. So Western blot is also quantitative test to determine the amount of protein in sample. Okay, depending upon what is the intensity of the color, you can find out how much amount of protein is present in your sample. Okay, so this is how you can see the protein of interest, the primary antibody, secondary antibody labeled with your enzyme. So this is substrate added. Substrate gets converted into your product, and product will give you light or give you a signal that is being present. Okay. So last part, let us quickly see about the application. So Western blot can tell us about the size and amount of protein which is present in the sample. It detects the presence of expressed protein in the recombinant clones. I have already told you about this. Disease diagnosis, yes, where specific antibodies can also detect a virus or a bacteria. Western blot technique is confirmatory test for HIV. It detects the anti-HIV antibodies which are present in the patient's serum, useful for detecting defective proteins like prions disease. Okay, usme it is directly prions are nothing but infective proteins. Yes, prions are infective proteins. So diseases which are caused by these prions that can be detected by Western blotting. Then you have definitive test for filled Jacob disease, Lyme disease, hepatitis B, and herpes. All these can be detected by your Western blotting.